my name is Amy, and today I'm going to be talking about a novel 28 amino acid peptide that we extracted from the Mexican blonde tarantula, and I'm going to be showing its anti-diabetic actions alone and also in combination with venom-derived exenatide. So yes, this peptide is novel and isolated from the Mexican blonde tarantula. It was researchers at Ulster University that had previously fractioned and characterized delta TRTX. So basically they analyzed the crude fraction and to see if it had any insulin secretory activity. Where my research is now focused on exploring the chemically synthesized version of this peptide. This is both in vivo and in vitro. Throughout this talk, you'll hear me refer to carboxylated and amidated. The carboxylated peptide is basically how the peptide is found in its natural form, but we've also went on to assess amidation of the peptide to see if it has any increase in potency or stability. And we've also went on to assess the effects of delta TRTX alone and also in combination with exenatide, which was extracted from the Gila monster. So basically the aims of this study were to determine the effects of delta TRTX on beta cell toxicity, proliferation, and also protection against cytokine-induced apoptosis. We also wanted to assess the insulin-releasing actions of delta TRTX alone and in combination with exenatide. We also wanted to uncover the PK profile of this peptide. This was both in plasma and also in pancreatic tissue. And we also moved on to mouse studies to assess its effects on glucose disposal and satiety. Again, these studies were carried out alone and also in combination with exenatide. So for our methods in vitro, insulin release was determined in the Brindy11 cell line. So basically we incubated the cell line with the peptide at 6 different concentrations for 20 minutes, collected the supernatant and then assessed insulin content via radioimmunoassay. In terms of beta cell health, the peptide was incubated at 2 different concentrations, so the 2 highest concentrations with the cell line for 24 hours. So then we assessed cell viability with MTT. And we also assessed LDH release because this is very important when it comes to insulin secretory activity. We also assessed proliferation using the Ki67 staining, and also apoptosis was assessed using tunnel staining. So for our methods in vivo, everything was carried out in healthy mice. So for food intake, we fasted mice for 16 hours. We then injected our peptide at 3 different concentrations. This was alone and again in combination with exenatide, which was carried out at a lower concentration of 2.5. In terms of blood glucose levels, the same experimental design was carried out. Fasted mice were injected with the peptide at 3 different concentrations, again alone or in combination with exenatide, and blood glucose was monitored for 60 minutes. And in terms of the PK profile of delta TRTX in the pancreas, we basically injected delta TRTX at 10 mg per kg, and then I collected plasma via cardiac puncture every 2 hours and also collected the pancreas every 2 hours. So in terms of our results, as we can see, the peptide, both amidated or carboxylated, had no effect on LDH release. So this means that the insulin secretory activity of the peptide is genuine. It's not causing the cell membrane to break open basically and cause insulin release. And in terms of the MTT, it had no effect on mitochondrial activity. So this would suggest that the peptides did not compromise beta cell health. So in terms of the effects of delta TRTX on beta cell health, this is in terms of survival and growth. So whenever we exposed the Brindy11 cell line to a mixture of cytokines, we did this in combination with the peptide to see if there was any protective action. And as we can see, the peptides protected quite significantly against cell death when we compare it to our cytokine mixture alone. And in terms of beta cell growth, we can see that the peptides significantly increased cell proliferation when we compare it to our media alone, and the actions are pretty similar to that of exenatide, which we would deem a gold standard for beta cell growth. So in terms of the effects of delta TRTX on insulin release from the BRIND11 cell line, so 1.1 millimolar glucose would be deemed as hypoglycemia. And as we can see, the peptides do not have any significant effect on insulin release. This means that the action of the peptides are glucose dependent, and therefore there would be no risk of hypos if you were to take the peptide. When we increase the concentration of glucose up to 5.6, we can see that the amidated peptide significantly increased insulin secretion at the highest concentration when we compare it to our glucose alone control. In terms of 11.1 millimolar glucose, this would be deemed as pre-diabetic. And as we can see, the trend continues with the amidated peptide significantly increasing insulin secretion at 10 to the minus 6 and also 10 to the minus 7. And this dose dependency carries on into our 16.7 millimolar glucose, because as we can see, the amidated peptide significantly increased insulin secretion, at 10 to the minus 8, 10 to the minus 7, and also 10 to the minus 6. So we then went on to test the effects of these peptides on satiety in mice. So everything is compared against our saline alone control and also our exenatide. So as we can see, the carboxylated peptide had no significant effect on food intake over the 180-minute time period, and both peptides were significant against exenatide alone, so the mice ate more. But when we look at the amidated peptide, we can see there's a significant effect at 150 minutes and also 180 minutes. This trend continues when we increase the dose up to 25 nanomoles per kg, because as we can see, the amidated peptide continues to significantly reduce food intake at 60 minutes, 90 minutes, 120 minutes, and also 150 minutes. But we see no significant effect with the carboxylated peptide. And again, when we use the highest dose of 250 nanomoles per kg, we can see that the amidated peptide continues to significantly reduce food intake at 180 minutes, 150, 120, and also 90. And again, the carboxylated peptide had no significant effect on food intake when we compared against our saline alone control. But it seems that we have the opposite effect when we test this peptide on glucose homeostasis in mice. So as we can see, there's no significant effect at 25 nanomoles per kg. This is our exenatide alone control, and it acts as expected with significantly reducing blood glucose levels. But when we increase the dose to 75 nanomoles per kg, we can see that the carboxylated peptide actually significantly reduces blood glucose levels compared to glucose alone at 30 minutes and also 60 minutes. But the amidated peptide has no significant effect. However, in terms of our area under curve, so under here, we can see that the amidated peptide nor the carboxylated peptide has any significant effect against our glucose alone control. But when we increase the dose to the highest concentration at 250 nanomoles per kg, we can see that the carboxylated peptide is significant when we compare it against our glucose alone control, as it significantly reduced blood glucose levels at 15 minutes, 30 minutes, and 60 minutes. And so we then went on to uncover the PK profile of delta TRTX in mice. So this is done both in pancreatic tissue and also plasma. So as we can see, in terms of the half-life of the peptide in the pancreas, it's 2.15, and this lines up nicely with the data that we got from the plasma, because it also has a half-life of 2.169 in the plasma samples. But we see a difference in Tmax. So in the pancreatic tissue, the Tmax value was calculated at 4 hours, whereas if we calculate it in the plasma, it takes 2 hours for the peptide to reach its highest concentration. So these are just some representative images of delta TRTX in pancreatic tissue. So this is our vesicle alone. So this was saline. And as we can see that the way that this kind of is lit up here, we can see the distribution of the peptide based on its molecular mass. Whereas if we look here, we don't see the peptide present. So based on this data, we were able then to go on and further characterize the distribution of the peptide at 2, 4, 6, 8, and 12 hours. And as we can see that the peptide is present right up to the 12-hour mark. So it would have been nice to further go on and see if it was still present after 24 hours. We then went on to assess the effects of delta TRTX in combination with exenatide on insulin release. So as we can see, there's no significant difference calculated between the peptide in combination with exenatide when we compare it to exenatide alone. But interestingly, when we look at delta TRTX right down to 10 to the minus 12 in combination with exenatide, we can see that it significantly increases insulin secretion compared to our 16.7 millimolar glucose alone. And the same can be said for the amidated peptide. When we administered the peptide in combination, we can see that it's significant right down from 10 to the minus 12 in comparison to our exenatide alone. So whilst it's not significant against exenatide, it is still quite interesting data. So then we went on to assess the effects of delta TRTX in combination with exenatide on food intake and glucose disposal. So we brought the exenatide dose right down to 0.25 nanomoles per kg. And as we can see, delta TRTX has no additive benefit effect when we compare it against our exenatide alone. And this can be seen with the AUC as well, that there's no more glucose-lowering effects in combination with exenatide than compared to the exenatide alone control. However, excitingly, when we tested this in its effect on food intake, we can see that the peptide in combination with exenatide significantly reduced food intake when we compare it against our exenatide alone control, and it's actually significant from 30 minutes right against our saline alone control. So this would suggest that there is additive benefit in giving this peptide in combination with exenatide on satiety in mice. So just to summarize the results, delta TRTX displayed no toxicity in beta cells. It enhanced beta cell proliferation and protected against cytokine-induced apoptosis. It displays glucose-dependent insulinotropic actions in the Brindy11 cell line. It moderately improved glucose homeostasis and induces clear satiety in mice. The peptide had a plasma half-life of 2.1 hours and a half-life of 2.1 hours also in the pancreatic tissue. So the peptide did not enhance the insulin secretory actions of 10 to the minus 8 molar exenatide in the Brindy11 cell line. It also did not improve glucose disposal when given in combination with exenatide, but it did clearly augment the satiety-inducing effects of exenatide. So just to conclude, delta TRTX is a tarantula venom dry peptide with a biological action profile of therapeutic interest for diabetes and obesity. This is both alone and in combination, so with the GLP-1 receptor, i.e. exenatide, signaling. So I'd just like to acknowledge my 3 different supervisors, Professor Victor Galt, Professor Stephen McLean, and Professor Nigel Irwin, and also Dr. Diego, because I wouldn't be able to do the mass spec without him. Also Dr. Neil Tanday and Gareth Orr and Diabetes UK for funding my research.
Thank you very much, Amy. So are there any questions from the audience?
Yes, I have a question. Jacob Fröhlich here. Thank you very much for your presentation. I was just wondering, when was this tarantula venom protein discovered, or did you also work on the discovery?
No, so it was discovered in 2015. So they had went on to characterize it. So my PhD focused on basically looking at the synthetic version. So I hadn't started my PhD yet when they'd already found the peptide.
All right, thanks.
Directly regarding that, I have also a question. Do you try, or will you try to change the amino acid sequence of the peptide, or are there any studies about to make it maybe even more potent?
No, because based with this tarantula peptide, it forms the inhibitor cysteine knot structure. So if we were to change the amino acid sequence, we would sort of risk maybe losing that structure. So that's why we just did a amidation at the end of the amino acid sequence to see if it had an effect on potency. But we haven't explored any further on trying to basically change the amino acid sequence.
Okay, thank you. Yeah, the next question is from Perry.
Hi, maybe I missed this in your talk in the very beginning, but why did you choose exactly that peptide? Did you do any pre-screening or high throughput screening or anything, or molecular modeling in silico?
No, so just basically the work that we do is very, very crude. So we basically take a crude venom, separate the different fractions, and if those fractions have insulin secretory activity, then we'll go on to further characterize them and get the amino acid sequence and then test it synthetically. So that would be like the only sort of screening that we would do.
All right, thank you.
Okay, are there any further questions? Yeah, the next one from Eric.
Thank you for the talk. I have a question regarding the potential application of this. As you said, the insulin isn't really affected much by it in comparison with just the exenatide. How high do you think are the chances that this peptide will be developed into a additional drug that can be used in combination with exenatide to reduce that satiety that you showed?
So I basically think that I wouldn't be able, so we carried out a long-term study with delta TRTX in combination with exenatide. So I feel that when we look at it on a very micro level with healthy mice, it's hard to say how effective this would be. I think in terms, as we know, obesity is very, very closely linked with the development of type 2 diabetes. So I think the fact that we're seeing a drug that can significantly reduce food intake is very, very has its advantages. But I don't think until we're able to look at all the long-term data of looking at this peptide in combination with exenatide in diabetic mice, I feel like only then we'll be able to comment on what could be its possible applications.
Okay, thank you.